The thing that's clear to us and what we've really seen in analyzing these products is there's not just 7-hydroxy that's produced. There's other unknown substances, and we still haven't figured out what they are yet. That's a big
Yeah.
project that we're trying to determine now because, first of all, we have no idea about the safety of 7-hydroxy in humans as an isolated compound. We do know that it has this danger potential, opioid addictive potential. And unfortunately, with the way that medical reporting goes and everything, poison control centers and whatnot, that data is so lagging what's really happening out in the real world.
Welcome back, everyone, to the Smarter Not Harder podcast. I'm playing the role of host today, and it's Boomer Anderson. We're back with another fascinating discussion around those topics which help us live smarter, not harder. And of course, if you're enjoying this podcast, I want you to smash that like button on YouTube. Please click subscribe. Or if you're listening to this on Apple Podcasts, Spotify, or wherever, leave a 5-star review because every one of those reviews helps. So today, I'm getting to have the second conversation. Actually, I've had many conversations over email with one of my favorite researchers in this health space, and it's Dr. Christopher McCurdy out of the University of Florida. And Dr. Chris McCurdy is professor in the Frank A. Duckworth Eminent Scholar Chair, Associate Dean for the Faculty Development, and the Director of the University of Florida's Translational Drug Development Core. He's a medicinal chemist by background, trained pharmacist, and somebody whose research I've followed for a long time, particularly around the subject of kratom or kratom or kratom as it's pronounced in certain areas of the world. And Dr. McCurdy and I would call this episode an exploration into various different compounds and plants, particularly around those ethnobotanical plants. We eventually hone in on kratom, but we talk specifically about Salvia Divinorum. We talk about this neuropeptide FF briefly. We get into discussions around cocoa leaves and so many more things. Once we hone in on kratom, we go into the mechanisms of action of kratom. And for those of you who are unfamiliar with kratom, or you may have heard of it because you've seen it at a gas station, your friend has talked about it, maybe somebody's using it that you know for energy to step down off of opiates, we get into some of the reasons why kratom may work for all of those different use cases and why kratom may actually be serotonergic in certain cases. We talk about specific alkaloids of kratom, specifically 7-hydroxymitragynine, and why that may play a role in opiate use, but also some of the concerns that Dr. McCurdy has over products being released to the market now. And then finally, we wrap up with a discussion around the future of kratom and kratom research. Again, fascinating discussion. So I don't really need to beat the bush here in the introduction. Let's get on to my conversation with Dr. Chris McCurdy. Dr. McCurdy, this is round 2 of a conversation between me and you, but this is the first time we've had you on the Smarter Not Harder podcast. Welcome to the show.
Thank you, and thanks for having me. It's good to see you again.
Yeah, absolutely. I hope you're enjoying beautiful summer in Gainesville. I'm glad to see that at least the air conditioning must be working today. So
yeah, today it was not last week or some time ago, but we're back in business. So.
Dr. McCurdy, just for, and of course we'll read your bio as part of the introduction, which will be recorded separately, but I want to dive a little bit into this whole fascinating field that you exist in of pain management, and to really understand, of all of the subjects that somebody wants to study in academics, how did you arrive at pain management when you were going through your various degrees?
Yeah, it's a great question. And I think pain management and natural products in general sort of found me. It wasn't the goal that I set out with originally. Originally went to pharmacy school to be a retail pharmacist and just practice in that setting. Along the way, I got very interested in research and started working with a faculty member in medicinal chemistry, doing chemistry research. And he said, you have a good knack for this. You might want to think about doing a summer internship. And so I went to the University of Georgia for summer. Didn't think much of it, loved it, had a great time there. Worked on compounds really for anti cancer at that point, and then returned to pharmacy school for my final year of pharmacy school and was in clinical rotations at that point, doing emergency medicine, intensive care medicine, just really challenging situations. Obviously, in those situations, you see a lot of people in pain. And I was fascinated by the fact that we really have poor pain medication choices. We have the hammer of the opioids, and then we have the nonsteroidal anti-inflammatory drugs, really, that encompass everything from ibuprofen to aspirin. And those are really the choices that we had at that point in time, at least when I was in school. I used to joke around. My father's a pharmacist too. I joked around with him that they only had willow bark to chew on and opium poppies to chew on back when he was in pharmacy school. But we have come a long way. And all kidding aside, to the pharmaceuticals that are available. But I started then into graduate school. I really had no intention of going to graduate school. It's a long story of how I ended up getting out of pharmacy from a practice perspective into going to the research side. And in grad school at University of Georgia, I ended up at there. And to be honest,
Just a quick aside here,
yeah.
Dr. McCurdy. Bulldogs or gators? You put yourself in a precarious
I,
position,
I,
right?
I learned a lesson very early in my career. My first job in academia was at the University of Mississippi, so the Ole Miss Rebels. And one of the faculty members there gave me a ticket for the Georgia basketball game. So Georgia was coming to Oxford for a good SEC matchup, and I show up with all my Georgia gear on. And I didn't know that this faculty member had two extra tickets next to the seats that I would be in. And he just looked at me in utter disgust and disappointment and says, "Son, you do realize who's on your paycheck?" I said, "Lesson learned." And so, yeah, I'm a Bulldog at heart, and I absolutely love the Gators now. And just really, from a baseball standpoint, we're big baseball fans, and we have season tickets to attend the baseball games, and I love it. Love to get to see all the different SEC teams come through here. Football is a little bit harder pill to swallow, but the way Georgia's been playing and the way Florida's been playing has made it a little bit easier in the past few years to be a outspoken Georgia fan. But yeah, I know who's on my paycheck, and I know what colors to wear and what emblems to wear in the right places these days. So that's a good
My brother
aside.
is a Georgia guy, but I interrupted you there.
No, that's
You're
fine.
in
That's
grad
fine.
school as
I
a Bulldog, and you eventually made your way over into the research side. How
Yeah,
did that
and so
turn?
that's where I started graduate school. I actually moved to finish pharmacy school, moved to Georgia, took my licensing exam, and became a licensed pharmacist in the state of Georgia, and still worked part time here and there. But I started on the PhD side of research and working toward that. Yeah. So the project that I had for my dissertation was really looking at a natural product from Indian tobacco. It's called Lobella inflata. And it was American Indian tobacco, not cigarette tobacco that we think of in the southern coastal areas. That type of tobacco is very different. This did not have nicotine in it, but it was used as a respiratory stimulant, and it did interact. These compounds still interacted with the same receptors that nicotine interacts with. And so we were really interested in understanding that. And this is where part of the pain situation started to find me. We were working on smoking cessation. We were working on a premise for Alzheimer's disease treatment as well, where the cholinergic hypothesis had emerged in the 1990s that smokers, for the majority of smokers, did not develop Alzheimer's disease. Now, it's a chicken and egg question because do they live long enough to develop Alzheimer's disease was that hypothesis. Yet most of the treatments that we still have available today are based in that sort of cholinergic hypothesis area. So we're studying this, and as I was becoming more and more of an expert on nicotine receptors and understanding the nicotinic system in our bodies, there was a discovery made at the National Institutes of Health where there was a compound isolated from a poison dart frog, and it interacted exclusively with nicotine receptors in the body. And it was an incredibly potent analgesic. And so nobody had seen this connection before. Some people talk about nicotine being able to help with pain and these types of things, but this was a really big finding, that this material that was isolated from this poison dart frog skin was really a painkiller. And it wasn't an opioid painkiller, but it had near the same potency as opioids. And the caveat again comes into the fact that it interacts with nicotine receptors, and nicotine receptors are not just in our brains, but they're throughout our body, particularly in our cardiovascular system. And so there was issues around developing a pain medication targeting those receptors that wouldn't affect blood pressure and heart rates and those types of things. But anyway, Abbott Pharmaceuticals actually took the lead. Now they're AbbVie, but took the lead on developing some compounds around this, and ended up publishing a paper with a compound that was 80 times more potent than morphine, working just through this nicotine receptor. And anyway, we kept doing our work. I was really starting to become interested in the fact that we could potentially find analgesics that were more potent than opioids, that didn't have that opioids. So this compound was really inspiring to know that we could potentially find other mechanisms to treat pain just as effectively as the opioids do, but not, hopefully not have the liabilities of addiction and other serious side effects that the opioids carry. I was fortunate enough to finish my degree and be offered a postdoctoral position, like a fellowship position, for 3 years at the University of Minnesota. And I worked with one of the world's foremost opioid chemist experts. And so I got to work on opioid chemistry directly with naltrexone, oxycodone, and those types of molecules. So we were looking for potential treatments for opiate addiction, potential new medications to really block the effects of opioids and help reverse overdose and those types of things. So I started to become not only this sort of non-opioid expert, but I started to become an opioid expert. And then I finished with that part of my life and started at the University of Mississippi as an assistant professor, and was looking for a first project. And I started with some other projects that I thought would be fun to just explore, some of which are still going on today that I thought would never amount to anything, but have been fun and still going on. And so we're working on those as alternatives, looking at alternative pain pathways or ways to modify opioid analgesia so that you can do what's called opioid sparing. In other words, use much less opioid so that hopefully you wouldn't develop tolerance as quickly, or you wouldn't develop the side effects as quickly either. So we work on a system called the neuropeptide FF system, and neuropeptide FF is a counter opioid peptide that's released in our bodies to be an anti-opioid, where it may help us from developing tolerance, side effects from our own endogenous endorphins. And this could be a whole story on itself, and we won't go down that rabbit hole, but that's a project that we still are working on. That led us into being at the University of Mississippi. We had the
All right, now, second sponsor for the day is Health Optimization Medicine and Practice, and it's our 501c3 nonprofit. Most people don't realize it's a nonprofit, and we deliberately structured it that way so that we can make a huge effect on this current healthcare system. And what's our objective there? Well, it's to establish a specialty in health within the existing healthcare system. And if you want to get involved, let's say you're a clinician, let's say you're a doctor or a non-doctor healthcare practitioner looking to practice health and give your parents, or patients rather, maybe parents too, access to health optimization, well, you can head on over to homehope.org and you can check out one of our 7 foundational modules. Our clinical metabolomics module has a cohort that meets at least twice a year, and that allows you to make a lot of learnings with 20 of your soon-to-be best friends in a weekly class-style format around topics like clinical metabolomics. The next one starts, depending on when you're listening to this, September 9th, 2024, and we have them 2 to 3 times a year. So please head on over to homehope.org, learn more about those modules. Please ask questions. You can send questions to info@homehope.org. And if you're interested, use the code podcast10. It'll get you 10% off your next learning adventure. Let's get back to the show.
The National Center for Natural Products Research there, and part of the USDA there as well. And they've long held the National Institute on Drug Abuse Marijuana Farm. That was the only federally legal farm for years and years. And so there was a lot of experience with plants there, medicinal plants. We started working with another faculty member there, Jordan Zavioni, who is a natural products chemist as well. But we started working on Salvia Divinorum. And Salvia Divinorum,
Wow.
was magic mint, right? And so
Yeah.
this was one of the most potent hallucinogens known, and nobody understood how it caused hallucinations. So everyone thought it had to be serotonin mediated, just like we would see with normal hallucinogens at 5-HT receptors, serotonin receptors. And surprisingly, it didn't interact with serotonin receptors. And then Brian Roth at the University of North Carolina, Chapel Hill, has the psychoactive drug screening program from NIH, from National Institutes of Mental Health, that sponsors that program. And he screened this compound through all the drug targets that he had. And it came out to be one of the most selective kappa opioid receptor compounds ever.
Huh.
And so the hallucinations were through the kappa opioid receptor. And then they were able to prove that this was there. We did work. We were funded with the first NIDA grant I received was actually on that project. We got funded at the exact same time that Brian Roth's lab got funded to study this as well. It was a fun time because the compound itself is not an alkaloid. It's actually neoclaridine diterpene. So it's a carbon and oxygen hydrogen molecule. It totally debunked everything we know about what an opioid should be and how a compound should interact with opioid receptors. It did not have a nitrogen. And so that was really the interest we had, is how is this molecule now interacting with a classical opioid receptor, and why so selectively? And we wanted to understand that. So we started moving down that road. Kappa opioid receptor agonists are interesting for a couple of reasons. They were thought to be the holy grail for pain medications because when they were studying these in animals, the animals did not have any interest in self-administering these compounds. Like, they will self-administer heroin or morphine, or they'll learn to take those because they give them pleasure. Animals did not self-administer these at all. And it got to the point where you were getting equal analgesia to morphine and those types of things. They developed very selective compounds for a kappa opioid system. They went into human clinical trials, and people took one dose and said, don't ever give me that stuff again, because what was learned was that the kappa opioid system causes dysphoria instead of euphoria. And so people had no interest or no desire in taking that. And so it's no surprise once that was learned as to why the animals were avoiding it as well. And just as a funny side note, while we were growing Salvia divinorum out at the field station at the University of Mississippi, it kept disappearing. Leaf were disappearing off of the plants. And we thought someone found out where we were growing it and was stealing it. So we set up trail cameras, and it turns out deer were out eating these plants. And so deer hallucinating out in Mississippi.
But it's, as you said about it being a kappa opioid receptor agonist, this idea of dysphoria, I guess, maybe doesn't apply to deer.
Maybe. And so it was just a funny aside, but there are a lot of people that were interested in Salvia divinorum and still interested. I know there's still some people that are interested in whatever that sort of feeling is. It is a plant that's used by the in Oaxaca, Mexico. That's where it seems to have its ground zero origin. It's used by shaman there to really, when they're stuck with treating an individual, they will either give the individual the salvia and that individual then can go inward and then have these hallucinations and then present what the issue is with them to the healer. And sometimes if they're really stuck, the healers would take it themselves to see if they could find the answers from the spiritual
Mm.
world. So just a really cool plant. We were the first ones to publish that it had analgesic effects as well,
Mm.
very short-lived analgesic effects. And then there was some interest in, could kappa make a resurgence if we could restrict it to the peripheral nervous system and not have it get in the brain.
Yeah.
And there's a lot of stuff going on. But when I was funded for that project from NIDA, and NIDA asked me to come and present on naturally occurring analgesics at a meeting. And so as I was preparing that talk, of course, you start with the willow tree and the willow bark and aspirin and morphine and the morphine poppy. And as I was going through and finding all these different plants that I never realized had analgesic properties to them, lo and behold was Mitragynine speciosa or Kratom or Kratom. And I looked at it and was just blown away by the different sort of alkaloidal template structure that the chemicals had within the plant. Started really digging in and reading what literature existed. And there was a really great paper from SmithKline in French where they took mitragynine, the major alkaloid from the plant. They actually took it into preclinical development and were going to make it into a pharmaceutical back in the 60s. They did the most complete work. That paper was published by the scientist was named Mako in 1972 when that paper came out. And to this day, it's still the most complete and thorough study of mitragynine I've ever seen in one document. But they did toxicology testing. They did all kinds of studies in different species to really look at, could they develop this further. And what they found was it had analgesic potency comparable to codeine, which
Mm.
codeine is very comparable to some of the nonsteroidal anti-inflammatory drugs in terms of its analgesic potency. Here you have a compound that was probably working through opioid receptors, and that was the assumption at the time, although this predates our knowledge of receptors when that
Yeah.
study was done. So they used an opioid antagonist that could not reverse all the effects. Turns out the opioid antagonist that they used at the time was not a very good opioid antagonist.
Mm.
And later on, people have been able to reverse the analgesic effects just with naloxone or some more pure opioid effects. But we'll come back to that discussion because we now have published some serotonergic and adrenergic mechanisms of pain transmission with mitragynine, as well as the opioid piece that's there. And that takes us into a different direction. We'll get there in a little while, but that's really where we started to go. At that point in time, it looked like the world just dropped this compound and this plant interest in it. And I started to find out there was a group in Japan in the 90s that was working on trying to develop analgesics from 7-hydroxymitragynine and see if they could come up with something that was more potent than what's out there now and less addictive. That group has stopped developing their work, but I'm not sure if they've stopped or not entirely. But we saw this opportunity and this door open for us to start a new project looking at new classes, chemical classes of potential analgesics from this plant. But as we kept reading about it and kept understanding what traditional use was of kratom, as it's called in Thailand, they
Mm.
would chew the leaf in Thailand, freshly picked off the tree, and just chew it. In Malaysia, they brew it into a tea. It's really a decoction. They spend about 2 to 3 hours boiling the leaf material into a aqueous extract, essentially, that they
Mm.
drink like a tea then throughout the day. But again, fresh leaf material. But it was all for energy. It was all to stimulate their ability to work in that tropical environment outdoors, mostly outdoor laborers. But interestingly enough, many of those individuals use raw opium. And when they would run out of access to raw opium, they would actually just increase the amount of Kratom they ingested, and they would stave off withdrawal symptoms and still get somewhat of the benefit of the opium, not as much, but there's just a ton of anecdotal evidence out there from Southeast Asia that this was helpful. And then later on, there was some reports where they were able to wean these opium addicts off of opium altogether. And that's where the sort of final light bulb went off for us, was to say, hey, we've been thinking about trying to develop analgesics, but maybe we should follow what's really out there and look at this as a potential treatment for opioid use disorder. And come to find out in different studies and surveys and things that we've done, and others are now funded in some of these areas, but there's use in alcohol use disorder in helping people curtail their alcohol intake. And also, we and others have shown efficacy in stimulant use disorder too, helping people get off methamphetamine or reduce their consumption of cocaine and methamphetamine, alcohol, and then obviously the opioids. And it almost sounds too good to be true. Is it a potential anti-addiction medicine that could work? And I say that because we filed a patent back in, I think, 2008 or some neighborhood of there, to do exactly that, to cover all the addictive space. And the US Patent Office came back with the most rejections that the University of Mississippi's Tech Transfer Office has ever seen. And they said, we're not going to try and prosecute and do any of this. That's where we left it. Now it's like hindsight, right? Starting to show all these great activities against other addictive substances, not just opioids. Anyway, that's a long story of how we got from doing these things to there. And also along the way, we work on a totally different project that I was drug into on sigma receptors, which were thought to be opioid receptors way back in the day. And many opioid chemists and pharmacologists died in trying to figure that out and understand how it was related to opioid receptors. But turns out it's not at all an opioid receptor. It's its own unique gene, but it happens to be involved in pain regulation as well. And so we have a diagnostic for nerve damage that's in phase 2 clinical trials that can pinpoint nerve damage locations that pain is originating from, which helps physicians guide treatment much greater to that specific site. We've been able to influence some physicians to actually go in and interrogate an area more thoroughly with MRI. And we've published a couple case studies now where we've cured, quote unquote, cured people of chronic pain because they were able to go in and do some sort of physical manipulation around that area to fix it. So
you're talking here about almost a diagnostic tool to pinpoint the sites of things like neuropathy and a few other nerve damage related disorders. So it's fantastic.
Yeah, so it's pretty cool. There's a lot of interest in there, and then things come full circle because this target is also in clinical development in Europe for Alzheimer's disease. And so we're back to where I started in that original sort of cholinergic hypothesis of looking at neurodegenerative disorders, not just pain and addiction anymore, but doing some other things along those lines. And it's really pretty cool, all the places that we're going. And the latest thing, which I know I didn't share with you before because it wasn't happening, but we're coming close to finalizing an importation source for coca leaf. And we want to start working on coca leaf.
Really? It's scheduled too, right?
Yeah. Okay. So I have DEA license and can do the research. The issue is getting the materials legally imported. And so we're working on that with some companies right now. And hopefully we'll have a cool project going pretty soon to start mining the alkaloids out of the coca leaf and see what got thrown out with the bathwater when cocaine got discovered and the rest of the plant totally lost. But again, another ethnobotanical story that you can, or ethnopharmacological story that.
Pardon the interruption, but I want to talk about one of our two sponsors today for this podcast, and the two sponsors for this podcast in general. The first one is Transcriptions. And Transcriptions, what we do is we produce nutritional formulations. And what we mean there is we're producing something for an effect in the form of a buccal troche. And in this podcast, you'll actually hear about things like metabolism, cytochrome P450 genes. And one of the great use cases of a buccal troche is to bypass first pass metabolism. So if you're interested in perhaps unlocking stress-free states, maybe getting some more sleep, or focusing for 4 to 6 hours at a time, go over to troscriptions.com, check out one of those formulations, and use the code POD10 to get 10% off. Let's get back to the show.
You can follow where people chew those leaves to get energy, and they don't have addiction problems. There's so little cocaine in that leaf material that they absorb it, but it's almost like microdosing. And so it's the next sort of chapter of where we're hoping to head.
Dr. McCurdy, I want to just first, there's so many interesting topics that you and I could cover. I didn't realize you were very involved in the research around Salvia Divinorum, and how, and that compound in particular or plant is fascinating and comes up from time to time in the psychedelic renaissance. There's also probably some questions that could go around ibogaine and your thoughts there, but just focusing on something that I came to know you for, which is around Kratom or Kratom, depending on who you talk to. And it's a plant that I had some exposure to when I lived in Southeast Asia, but kept hearing about it more and more, and would love to just understand from you. And you mentioned something earlier about potential serotonergic properties of Kratom, and just how does the plant work in humans? Maybe we can get into some of the mechanisms of action, and what do we know about how the plant works in humans?
Yeah, so it's a really good question, because it's not a very long conversation as to what do we know about how it works. We really don't know a lot right at this point. We do know, and part of the funding that we had from the National Institute on Drug Abuse was really to go in and isolate out as many compounds as we could from the plant, purify those, study those in a similar fashion to like what I mentioned before when they screened Salvinorin A, and they found that against hundreds of targets that it was just interacting with this opioid, kappa opioid receptor. We did that same sort of screening campaign with each of the alkaloids that we were able to isolate and try to understand what is going on with the mechanism. Of course, everyone was focused on opioid and opioid alone, but as a medicinal chemist and just looking at the structure, I said this has got to have serotonin activity. It's an indole-based alkaloid, and that indole ring has a 2-carbon separation to a nitrogen that's protonatable, and so that's the tryptamine type core scaffold. And so we were not surprised when we first screened Mitragynine that the data that came back, we saw that it had affinity at mu and kappa opioid receptors. We expected to see that. And then I think what kind of surprised us was seeing that it had pretty selective serotonin activity. It interacts with the 5-HT1A receptor, and I'll talk about the pharmacologies that are believed to be associated with that. And also it interacts with the 5-HT2B receptor. And so the 5-HT1A receptor has been reported to induce respiratory depression. So this made us come to think about a lot of the reports where people use Kratom or Kratom. A lot of the reports where people say it's safer because you don't get the respiratory depression, and you don't have as much respiratory depression associated comparative to an opioid. Could it be that this 5-HT1A receptor activation is actually mitigating some of that through a pharmacological mechanism and not just a unique opioid mechanism, which is also something else that's been reported, because it has at the opioid receptor, Mitragynine is a partial agonist, so it doesn't activate fully like something like morphine would do. It only can reach about 40% activation of maximal, and it does not seem to cause respiratory depression in animals when it's given alone. And furthermore, it's what we call a bias agonist, and so In the signaling world, a compound interacts with the protein changes conformation, and it recruits other proteins on the inside of the cell to activate and start what we call second messenger signaling. A process that stops that is a protein called beta arrestin. And beta arrestin is recruited under typical opioid analgesic compounds will recruit beta arrestin, and mitragynine does not recruit beta arrestin. And
Mm.
this is why it's called a biased signaling molecule. And what is believed by several pharmacologists that these bias signaling events or beta arrestin recruitment, in other words, the non bias, actually are connected to the side effects. And the respiratory depression and the constipation is more a result of beta arrestin recruitment than not. So there's two possible mechanisms out there, right? There's this sort of signaling pathway that can be different than a traditional opioid, but there's also this polypharmacology aspect that we really believe more in from a standpoint. And I'll tell you why as we talk about some of the other alkaloids. And I'm spending a lot of time on mitragynine itself because it's the one that's been the most studied and the one we know the most about. And
Mhm.
that's just simply because it's the most abundant one and it's the easiest one to isolate and study. We also saw this 5-HT2B activity, which was concerning because that's an anti-drug target. This is
Mm.
known. This was known really discovered during the sort of Fen diet medications
Yeah.
where started to get cardiac valvulopathy, and people would have cardiovascular incidents, heart attacks. And so it was tied then to this 5-HT2B receptor activation, and it is a anti-drug target, so we don't want to target that, we don't want to activate that. However, mitragynine and the other kratom alkaloids seem to block that target, so they're not having that pharmacological effect that you would get with an activator or agonist of that target. So that was a good check the box. We're okay as far as the cardiovascular side goes for that piece. We were also surprised to see that it had alpha adrenergic activity. And alpha adrenergic receptors are on blood vessels. They're also in the brain. They're on nerves and in the heart tissue. And we know that these alpha adrenergic receptors are part of our fight or flight mechanism. It's where stimulants act. And we started to piece together this whole pain activity coupled with increased mood elevation, potentially through serotonin interactions, and also this energy boosting through probably the adrenergic system. And so it started to make sense from a pharmacological standpoint if we're activating all these receptors. And again, this is just mitragynine. We're talking about one molecule. So we went further, and as we isolated more compounds, different alkaloids, highly structurally related to each other, by the way. But as we start to look at the differences, and in some cases, it's just the orientation of a hydrogen on the molecule is pointing up instead of pointing down. And that changes the three-dimensional conformation of the molecule completely. And just to make a clear example, a compound called speciogynin, which is that isomer of mitragynine, where it's just got this
Hmm.
one change in it, it flips from preferring to bind to opioid receptors to preferring to bind with very high affinity to 5-HT1A receptor. And so it's the second and third most abundant. Painanthine is a structural precursor to speciogynin, which differs only by a double bond, and the pharmacologies are really identical. But the two of those alkaloids make up together a pretty significant portion of what's being exposed in the body as well. And so you're getting this high serotonergic activity coupled with these opioid like activities. And as I said, it prefers to bind with higher affinity to serotonin, but it still interacts with the adrenergic, it still interacts with the opioid system. And now you're starting to get to a point where it's like real estate, it's location. Where is that compound?
Yeah.
Where is it acting in the body? How much concentration is there? Is it acting preferentially over another molecule that could be present? And then as we started to dig through more of these compounds, sure enough, we found compounds that were very selective toward the adrenergic system. And so that theme has stuck with these alkaloids where they have opioid, serotonin, and adrenergic activity, which makes them very atypical from any type of compound that we've seen before and in nature other than ibogaine, but these are not like ibogaine. Ibogaine is
Yeah.
another dirty molecule that binds with a lot of targets, or the new more correct term is polypharmacologic. That sounds better than dirty. But
Yeah, this is fascinating because when I heard you speak about kratom, I kept thinking about ibogaine. And with kratom, you have, I have people that are, I would call them more acquaintances that report so many different effects, from like energy better than coffee to pain mitigation to just being able to relax. And you think to yourself, like, what the hell? This is like all report. And of course, there's sourcing issues and all this stuff. But it's just it was like hard for me to explain, or I couldn't make up any sort of reasoning for why this would exist. But now just hearing you talk about the mechanisms here,
yeah,
it appears that we have a lot of different alkaloids to talk about.
and every one of those compounds has analgesic activity, and we can reverse. So if we take a compound like speciogynin, which has the higher affinity at serotonin receptors, it has analgesic activity, and we can reverse that analgesic activity not with an opioid antagonist, but a serotonin antagonist. And
Mm-hmm.
same thing with the adrenergic side. We can see adrenergic mediated analgesia, and we can reverse it with antagonists at those targets. And so it becomes very fascinating that each of these molecules individually have unique pharmacology. And then you have to put your mind back together and realize that no one is taking these in isolation. They're taking them all in this combination product, if you will, of creating. And it's a complex, and I always refer to it as a complex symphony orchestra because you have so many different molecules playing all these different pharmacology, and depends on the dose, right? So a lot of people say that less is more in the case of kratom use, but people that are trying to get some sort of higher euphoric response, they're pushing the doses higher and higher because it doesn't give you that sort of instant euphoria that you would expect based on all the hype that's out there and based on where you can purchase it. It's not at the mainstream GNCs, or you can't go on Amazon and buy it, or Walmart. It's at gas stations and at smoke and vape shops, and you generally associate the products that are available there as not as mainstream. We have to take all of these things with those thoughts in mind, and really understand that this plant is incredibly complex, and putting all those molecules back together that we've studied individually is not even the story that you can do. So part of our grant was to find out which alkaloids get into the blood from ingestion of leaf, then
Mm-hmm.
take those alkaloids and reconstitute those into purified cocktails. So we could take each individual molecule that we know is getting into the blood supply, and then can we reenact that same formulation with purified material that down the road could be more of a pharmaceutical, or a very highly purified controlled product. But you can't. You've got to have the sort of magic of the whole symphony orchestra there. It's like it really is. It's like taking the whole symphony orchestra and just pulling out a few of the instruments and letting them play on the side. You miss a lot of the subtleties.
So you hear the people that are particularly involved in cannabis medicine, or sometimes even pipermethysticum or kava, they talk about the entourage effect, right? And
Right.
it sounds like we have a bit here with kratom. And maybe to go against the entourage effect, I want to just hone in on one particular alkaloid and just get your thoughts on it, because you see it pop up sometimes in, I don't know if it's a European pharmaceutical company or if they're technically a US pharmaceutical company. There's people out there studying 7-hydroxymitragynine. And I would love to get your thoughts around 7-hydroxymitragynine as an alkaloid itself, as potentially for an alternative to an opiate, but also there are products out there that are trying to isolate it, and what sort of the dangers are around 7-hydroxy.
Yeah, so first and foremost, we now know that 7-hydroxy is not produced by the plant. To the best of our scientific knowledge, it is a post harvest artifact that develops once the leaves are separated from the tree. Our group has reported on that. We've worked on the biosynthetic pathways with some of the best biosynthetic folks in the world at Max Planck Institute and Sarah O'Connor. We've been doing genetic studies here at the University of Florida as well. And the groups in Malaysia at the University of Science Malaysia actually just came out with a report very recently showing that and proving that 7-hydroxy mitragynine or mitragynine is not produced by the tree. And that's what we do know. We also know that it's generated through metabolism. And when we ingest, or let's say when we give pure mitragynine to an animal orally or IV, you get production of 7-hydroxy. And this is because there's a specific metabolite enzyme, cytochrome P4 350, cytochrome P450 3A4. I was trying to say too many numbers and letters at once. But CYP3A4 is present in the intestinal wall as well as in the liver. And so if you ingest Kratom product orally, you convert some of that mitragynine in the gut to 7-hydroxy, and then some of that mitragynine that makes it to the liver then gets converted there to 7-hydroxy as well. And when we've given it IV to animals, you get conversion just from the liver. So you don't have that gut conversion. So you see less produced when you give it IV. And so this was consistent with what was reported way back when in the literature, where they noted at SmithKline and French when they were doing these studies that the compound was more potent when it was given orally, which should have been a pretty dead giveaway that some sort of metabolite was being formed. But back in the 60s, 70s, it just wasn't as easy to figure some of those things out as now the technology is enabling us today. So those are the things we know. We also know that 7-hydroxy, from that big screen that we did against all these targets, drug targets, is incredibly selective for only opioid receptors. So it only interacts with mu, delta, and kappa, those three classical opioid receptors. In fact, I always say it's probably one of the most selective opioid compounds I've ever seen. Salvinorin A would be in that category as well, and it's only for kappa. But 7-hydroxy interacts with mu, delta, and kappa, all three opioid receptors. It interacts the most with the mu opioid receptor, which is the morphine receptor, the one that we're all familiar with causing our clinical pain target, and also our problems with clinical pain medications. And unlike mitragynine, where we talked about it being this sort of biased agonist, 7-hydroxy mitragynine behaves much more a pure opioid, and it does recruit beta arrestin. Now, interestingly, in there, you do get respiratory depression with 7-hydroxy, just like you do with traditional opioids. Now, the other piece of that puzzle is you're not getting that 5-HT1A activation with 7-hydroxy that you see with mitragynine, right? So it's not doing that polypharmacology either. So that it just throws a wrench into all these theories, but they're all still possible. But we do know, and one of the big questions that I had early on when we started researching this at all was, does 7-hydroxy have abuse liability because it's much more pure opioid? So we did do a study in rats, and we know that rats will train themselves to self-administer 7-hydroxy. They will not do it with mitragynine. And 7-hydroxy substitutes directly for morphine in animals that are trained to self-administer morphine, whereas mitragynine did not substitute. The abuse liability difference was pretty clear to us. So we published that, I think, back in 2019. That work has been followed up on by other group, actually from the National Institute on Drug Abuse as well, showing a very similar outcome with mitragynine. They did not do 7-hydroxy, but 7-hydroxy we think is by itself a potentially dangerous and addictive substance. It's concerning now because we're seeing companies put out products that are claiming to be 7-hydroxy isolate tablets or shots. The one good piece of news is that it doesn't cross the blood-brain barrier as efficiently when it's by itself as it does when it's in the presence of some of the other kratom alkaloids. But again, those are in mice and rats, so I can't say that's the same outcome in humans. But we've done analysis of some of these new 7-hydroxy tablets and liquids that are out there. It
Are they
appears
actually 7-hydroxy? Because that would seem to require a lot of chemistry and
so they are
the
definitely
places that you're seeing.
they're definitely being synthetically made because there's no way you could isolate that much 7-hydroxy. As we talked about, it's just not really present in the plant. You can, though, convert mitragynine, which is very abundant, what we call semi-synthetically, to 7-hydroxy. And we think what we're seeing with these products is exactly that. So they're taking a enriched extract, a mitragynine enriched extract, then chemically treating that to produce the 7-hydroxy as the main component of that extract instead of mitragynine being that main component. The thing that's clear to us and what we've really seen in analyzing these products is there's not just 7-hydroxy that's produced. There's other unknown substances, and we still haven't figured out what they are yet. That's a big project that we're trying to determine now because, first of all, we have no idea about the safety of 7-hydroxy in humans as an isolated compound. We do know that it has this danger potential, opioid addictive potential. And unfortunately, with the way that medical reporting goes and everything, poison control centers and whatnot, that data is so lagging what's really happening out in the real world. It's going to be that genie's out of the bottle. It's been out of the bottle for over a year now that these products have been available, and they just keep increasing in their availability. And we're seeing new products introduced. It feels like every week. I know it's not that frequently, but there are an increased amount of products coming out, which really more like pharmaceuticals than they are natural products anymore. Not really.
One of the things that Dr. McCurdy, one of the things that fascinates me about these products, and the reason why I double clicked there and said, are these products actually 7-hydroxy, is because a lot of these products are being sold, as you mentioned before, in gas stations, right? And places that you wouldn't traditionally think of as a place where you'd go to purchase a pure molecule, right? If you're buying 5-hour energy at a gas station, that's one thing. But you're going and buying something that is a very intense opiate, or forgive me if I'm not getting that term correctly, but I guess my concern here is the end consumer may not be as well educated. And how do you look at some of these and the combinations that are out there? And some of them have a lot more press around them than others, where they're combining it with a positive allosteric modulator, the GABA receptor. How do you even start to, I'm surprised you still have hair, frankly, but like, how do you start to look at these different compounds and say, okay, you have a great plant, but now these people are doing all these crazy things with it?
Yeah, it's human nature to fool around with stuff. Yeah, it's not surprising seeing what's happened and combination type products. There's lots of things being combined with kratom, from CBD, which is said to really enhance the chronic pain efficacy of kratom, to kava, which is said to help with the focus and the calmness and sort of a social lubricant, to other compounds like terpenes from cannabis and other nutraceuticals. Can't think off the top of my head right now, but there's so many things being put together because that's a lot of what we see in the dietary supplement space already, where we see these combination products of if this compound is good for you and this compound is good for you, or this plant's good for you and this plant's good for you, we'll make it easy for you and put them together so that you can just take it as it is. And we have no idea, especially when a substance has got psychoactivity like kava and a psychoactivity like the kratom itself, and psychoactivity like marijuana. There's not products that combine marijuana and kratom, but there's a tremendous amount of individuals that are doing both together. And there's things called KFC where they do kratom food and cannabis, and
I
there's all
heard that
kind
one.
of there's crazy stuff out there that people are doing or promoting, particularly if you follow some of the Reddit threads. But we just don't know, and we don't know about these individual plants themselves very much, let alone combining them with other plants, and then what the consequences could be. Because one thing that the general population and even some of us in the medical field don't really think about too often is all of this stuff has to get metabolized, eliminated from the body. So we're putting it in with no question, but the body's then getting totally confused as to how do I get rid of all this stuff, because that's the body's main goal, is to just get rid of anything that's not of use to it for energy, right? Yeah, it's very confusing. It's very concerning. We do know that, for instance, there is an effect. We published a paper last year where CBD does enhance the exposure of kratom alkaloids. So you actually get more sort of absorption. Is it blocking metabolism? Probably as well. We know that the kava lactones are metabolized through the same metabolic enzymes that the kratom alkaloids are metabolized through. And so it's a revolving door, more or less, in these metabolizing enzymes where it can only handle, an enzyme can only handle one substrate at a time.
Yeah.
And one substrate is going to have a preference over another. And so what happens is that revolving door, if business is good and steady, it can keep up with what's going on. But if someone's in the mall and there's a fire announced and everybody in the mall tries to get out that one revolving door, all of a sudden the smaller people are going to slip out first. And it's the same way with these metabolic enzymes, where they're going to build up and you're going to build up, accumulate compound in the body more than it can be eliminated. And that's when you can start to get into some adverse events or dangers with the way that things build up.
Dr. McCurdy, this is amazing. And if there's many different avenues this conversation can go, I want to, in the denouement of this conversation, go into a previous documentary I actually saw. You were in it that actually helped prevent kratom from getting scheduled. And you have that on one side, and you have all of these people using it to step down off of opiates. You have all these people using it for various health effects. And then on the flip side of it, you have a lot of these products that are being introduced that haven't been tested in humans. They're using 7-hydroxy, they're using other compounds, which we don't know exactly how they interact. How do you balance it all? And if you're sitting there today, how do you look at the future of kratom? Because at one point, this was something that was on the fast track to being scheduled. And now it's sitting in that in-between land with some potential danger on one side and a lot of beautiful benefits that should be accessible to anybody who needs it on the other.
Yeah, and I think it's a great question, and it's an unfortunate place that we're in at this point in time. But I think we have to contextualize what time frame we're talking about. So the scheduling first came about in 2016, and in 2016, I would even go as far as to say, even in 2021, major products that were available were leaf material products. So they were not manipulated into extracts. They were not made into concentrates, and certainly not into isolates. Isolates really started to appear in 2023. And so when you have a discussion around scheduling something in 2016 that was just leaf material, that does have a traditional use and anecdotal evidence of medical benefit and potentially harm, of course, when that's what we really want to understand is where is the benefit and then where does the benefit end and the harm begin. And what we've seen bear out over these last years is people that are using leaf material in a responsible way. In other words, they're using a small amount, they're not escalating their dose over time, seem to be able to manage the use in a responsible way and benefit from the plant. So it's also scary to think that we are approaching probably somewhere in the 15 to 20 million or more user range in the United States of this plant. Back in 2016, it was a fraction of that. And you can see how it's grown in popularity just by driving down the street now. You see neon signs next to the neon CBDs in many of the gas stations that weren't there even a year, 2 years ago. The thing that we have decided to do as a group of scientists concerned about this for sure and the changing landscape is really focusing on a paper we published a commentary that's called Not All Kratom is Equal. And right now, the way that anyone views products, it's all lumped under that one name, right? It's all kratom. Whether it is leaf powder or it's isolate product, they're all being called kratom products. It's really, in my mind, analogous to the alcohol industry at this point, because we can call alcohol, and everybody knows that alcohol encompasses this wide array of products from spritzers or seltzers or light beers all the way up to one Everclear 190 proof.
Yeah.
And the scary part is that you've got this variety of liquid shots out there that are being sold in gas stations and sold in vape shops. And these products, and by the way, there are kratom products now too, which we're just getting into studying.
Of course there are.
5 Hour Energy, and it's a perfect example. I'll never forget, we were driving up the interstate, up I-75 out of Florida, and stopped at a gas station to fill up in Georgia. And right next to the 5 Hour Energy was shots, right at the register counter. No, not under lock and key, not anything there. So you got to worry that people think they're grabbing a 5 Hour Energy and grab the wrong thing. But we're talking about some of those liquid shots are not concentrated extract. Some are 3x, but now we're getting up to ones that are hundreds of times potent compared to just a regular leaf material. And to the point you made earlier, someone just going in there and grabbing that as their first experience and just completely knocked off their shelf because it's like having a shot of 151 for the first time you ever try alcohol. It's your body doesn't even know what to do. and so this is the concern for us, is that the regulators and everyone else that's out there right now is looking at all of these products as kratom, when in fact they're not equal at all. And now we're trying to make some sort of plea to these Kratom Consumer Protection Acts that are going in to really stay true to the products that are benefiting people, not the products that seem to be out there and being abused and designed, for lack of a better word, to give you some sort of euphoric feeling. They're putting these liquid shots in tiny vials and saying there's 9-10 servings in there. Who's going to open a vial? And
are you going to take a teaspoon?
Yeah.
You don't take a teaspoon.
It's just absolutely gotten out of control in our minds. And my fear, and the other scientists' fear, is that it is going to push itself towards a scheduling event. And again, as everything is going to be equal in the eyes of the legislators right now, those new products that are coming out and these really potent isolates that are going to create problems. And again, that's going to lag for some time before we really see those. It's going to put an end to the whole thing. And then you turn access off to people who are legitimately using this, that really need it, that are really benefiting from it, and everything goes into a black market. And there's still going to be products out there. There's still going to be things available to buy. But now, instead of being able to get it at the vape shop or even the gas station, it's going to be gone from those places and turned into more of the illicit market that we see drugs being sold in. And it's just a scary thought to think it'll go that way. And that's what we're afraid of. We don't want to see people lose access to the leaf material, the way that it was intended to be used in more traditional manner than what's out there. So that's my big soapbox for now. We really are working hard to try and get attention of DEA, of National Institute on Drug Abuse, FDA, everyone that has a voice in the regulatory space and in the justice space to get these products that are, potentially very dangerous out of commerce. They should be studied like pharmaceuticals and not as some sort of dietary ingredient or natural product. So that's our big thing right now. We're working really hard to try and do that. Unfortunately, the way that the systems work, until there becomes a big problem and a bunch of people start dying, we're not going to see action.
Well, Doctor, just before we close off with our final question here, I want to thank you, because you seem to have, and I appreciate how you outlined your career and how you've come through these various plants and ethnobotanicals and stuff like this, because you've had this amazing career of going through and studying really difficult and controversial plants, whether it's Salvia Divinorum or Kratom, or it sounds like coca leaves are on the horizon. But the work you do is fascinating and just incredibly interesting. And so thank you. And
Thank you.
I do hope that the work continues and that we get some responsible entrepreneurs out there that are using these plants as they were originally intended in their native cultures. And perhaps that's the way that this can move forward safely, effectively, and without it having to be scheduled. I hope. Asterisks, right?
Yeah, I will just say in wrapping that piece up too, just real quickly, that most of the vendors that have been around for quite some time now are doing things in the most proper ways I think that they can. I have a relationship with many vendors, as you can imagine. We share our science with them. We try to encourage them to do best practices and types of things that we learn. But we also see a bunch of people, just like we did when CBD and still going on with Delta 8 and some of these things, but people that are just out and interested in making a buck. And unfortunately, they don't care at what cost that is to other folks' lives. They're just happy to profit in their own pocket. And I've heard rumor, and it's obviously it's hearsay and not something that is factual, but that's how us scientists get our hunches and move forward. But I've heard that one of the companies selling 7-hydroxy tablets is profiting around $4 million a week. That's a quick money grab when you think about it, without any thought of what the consequences are. To say there's some bad players out there, yeah, but I think the majority of the established vendors are trying to do the right things and get better and make it as legitimate as possible.
Well, I think I know one of the ones that you've talked to in the past. I won't give a shout out to him here just in case he doesn't want it, but he used to live in my homeland in Amsterdam. But Dr. McCurdy, the last final question that we ask every guest on this show. And again, I just want to reiterate my thanks for you spending time with us today and educating us. This is a very good deep dive into what we know and what we don't know about both kratom and so many other things. What piece of advice would you give anybody listening to this show to live smarter, not harder?
Live smarter, not harder. I think if you're gonna do things smarter, it's all about education. And that's been a cornerstone of where I started and where I continue to go and where I live on a day-to-day basis. It's learning more so that, quite honestly, I don't have to work harder. So I think it's reading. I think it's educating and opening your mind to things that may be controversial and trying to get a better understanding of that. See both sides.
Thank you so much, Dr. McCurdy, for being with us today. If people wanted to find out a little bit more about you and your work, where would you send them to?
So most easy way to find me is LinkedIn, of course, but the University of Florida College of Pharmacy website has a good link and presence there. I don't have my own personal website or anything like that. What we try to really communicate through LinkedIn, our discoveries and what's going on. That's really the best way to keep up with what we're doing. PubMed is another one that we should have a new paper on there tomorrow. In fact, it's always exciting and always things coming out. So just pay attention. But the University of Florida website does a great job of auto-populating the latest papers that are out there in the press, so you can find what's going on and what's happening with us.
Beautiful, beautiful. Dr. McCurdy, thank you so much for being here today. It's been a real pleasure.
Thank you. Thank you. It's always fun chatting with you, and I'm sure we've got other topics to breach in the future as well. So I look forward to.
I'm coming back to you on neuropeptide FF and probably some of the work you're doing on cocoa leaves, and you said ibogaine, so I have so many more questions. But thank you very much for coming on the show, and to everybody listening out there, have an absolutely wonderful and health optimized day. Wasn't that just a fascinating conversation? Dr. McCurdy and I could have easily gone into many different directions, whether it's been Salvia Divinorum, neuropeptide FF, or even cocoa leaves and some of the things that he's doing there. Of course, I'll have Dr. McCurdy back on the podcast at some point in the near future. And if you're listening to this and you say, well, wow, that was a fascinating conversation, well, smash that like button and subscribe on YouTube, or leave a 5-star review if you're listening to this on Apple Podcasts or Spotify. Now, one thing I just want to thank you, the listener, for going through here is that we did have a few interruptions. The Wi-Fi connection on Dr. McCurdy's side was a little uncertain, but we made it through and we had a fascinating conversation as a result. So thank you again for listening. For those of you who want to check out the show notes on this one, because there might be quite a few, you can head on over to transcriptions.com or homehope.org and you can access the podcast right there. Thank you again for your attention in this otherwise very distracting world. And thank you for delving into a very, very important topic with me on health optimization. Have a wonderful day. And don't forget to live smarter, not harder.